It is a great pleasure to be here in rainy Baltimore, and I want to welcome all of our folks who are dialing into this lecture. It's a great pleasure. It's my pleasure to introduce Assia Golizar. She is a good friend, and she's also the Vice President for Global Head of Data Science at Odysseus Data Services, a leading real-world evidence technology vendor. She is a professor of the practice and the director of the clinical research at the Odyssey Center in Northeast University. And Assia has more than 20 years of experience in life science research and medicine and industry, academia, and government settings. She holds a PhD in epidemiology and a master's of health sciences in biostats from none other than the Johns Hopkins University School of Public Health. She was supported in her postdoctoral research fellowship award with the National Cancer Institute's Division of Cancer Epidemiology and Genetics. She earned her medical degree from the Tehran University of Medical Sciences. And after receiving her PhD, she joined the faculty in the Department of Epidemiology at Johns Hopkins in our School of Public Health, focusing on cancer and diabetes epidemiology and applying evidence-based findings to strengthen public health infrastructure and policies. She then joined the pharmaceutical industry, where she has worked as a epidemiologist in therapeutic areas, leading an expert at Regeneron Pharmaceuticals, AstraZeneca, and Bayer, and has led and contributed to the integration of effective and efficient observational research strategy into the research and development, clinical development, and lifecycle management in different therapeutic areas, specifically in oncology. And then in 2018, she led the Odyssey Oncology Working Group, endeavoring to extend the OMOP CDM to support oncology use cases and advance large-scale observational oncology research. At her current role, she leads a team of data scientists, epidemiologists, and bioinformaticians focusing on epidemiological research and development and application of advanced and innovative analytics across a large global network of observational data. Well, that was a lot, but let me tell you why I invited Assia to come give this talk. Here at Johns Hopkins, we are really been excited and embracing the Odyssey data model. And what we find is it does such a great job of capturing medications and labs and procedures and diagnosis codes, but we're trying to go deeper. How do we get organ level data? How do we find new phenotypes? So we've created a lot of internal clinical registries, and we're always trying to extend the OMOP data model. We're trying to add new features into our model. And Assia has led one of the best efforts of extending the OMOP data model. So she's here to talk about how we can go to really start getting to much more granular measurements around our patients to be able to help us enable and accelerate biomedical discovery. Assia, thank you.
Thank you so much, Paul. And thanks, everyone. It's really a great pleasure to be here, pretty much being back where I grew up and spent most of my early adulthood. So as Paul said, I'm going to talk about specifically oncology. This is what I'm passionate about. Some people call it my first love. And I'm going to start sharing, but please, let's keep it conversational. If something comes up, please feel free to ask questions. I won't be able to see you. So Paul, maybe you can help moderate that or just
I can. So please ask questions in the Q&A or in the chat of the Zoom webinar, and I will be more than happy to interrupt Assia if the time is right, as part of the lecture or as soon as she's finished.
Can everyone see my screen?
It looks great.
Great. Okay, so today I'm just going to talk about the observational cancer research at large scale and what we've done, the journey we've gone through, and just give you an idea of how we see that and where we're going and where we are. So not surprisingly, I have to talk about my disclosure. This is my disclosure slide. Used to be working at Regeneron Pharmaceuticals. And very specifically, I want to spend a few minutes first, just talk about some of the challenges and limitations of large-scale observational cancer research. Why am I here and why are we talking about cancer differently? What the Odyssey community as a group has done in the past several years to overcome some of these challenges, where we are with that. And then I would like to just give you some of these offerings, what we call the oncology module and action. So not surprisingly, we are all a part of the Odyssey community. We are a community of scientists, clinicians, epidemiologists, informaticians, and you can name it. This number needs to be updated, over 3,000 collaborators around the world, different time zones, different continents. The data network within the community is around 331. That was the latest. I'm sure it's much more than that. Different types of data from EHR claims, also registry, different countries. And we're looking at over 8 million unique patient records within the data network. So as a community, the entire collaboration, like within this journey, we're focusing on open community data standards, standardized methodology that can be used for reproducing reliable and reliable evidence, and also development of tools that facilitate that. And we're doing that all to generate clinical evidence that can be used for different types of decision making, either at bedside or for policy makings or guideline development. So you might have seen this before, but I would like to just give you a little bit of like recap of what is it that we're talking about. The current type of like study approach that we do, a lot of like the work that I used to do when I was doing a lot of epi study was that I have a question, or someone comes to me with a question of, okay, what's the adherence of my drug in the data that I own? So I have a specific analytic, I have to put together an analytic method to look into the adherence of the drug, apply to my data. My data is only one of them. That creates a problem, right? So like if I want to do that, like for each of these application, I have to write a specific SAS script or R script. That means that it's not scalable. It's really not transparent. There's a lot of room for mistake. It's expensive, it's slow, and it's not something that we can do like routinely and at scale. So just think about it like different questions and different data. What Odyssey the Odyssey approach is actually different in terms that I have a set of tools and my data. So it can be adherence, and the adherence can actually be answered in a series of database, no matter where they're coming from, how they look like, as long as they're all standardized the same format, what we call my OMAP common data model. So thinking about the plugs, the moment you have like the plugs all look the same, all I need is only like one script that can be applicable, and it means the same. The data means the same across this. And then I can have like a series of tools from adherence, mortality, safety, prediction, and all that. At the same time, because everything is standardized, analytics can be remote. It can be sent to the data. I don't need to see the data. I know how the data is structured, what are the vocabularies, what is the model that maintains the data, so I can send the analytics to the data. The data analytics can be behind the firewall, so we're sort of ensuring patient privacy and a lot of issues around that. And more importantly, that enables us to do a research network. So the data is pretty much standardized analytics, it's sent to standardized data. That can be like a combination or a linkage of different data within a research institution that can come from like commercial entities, like commercial claims, and also different partners. We can pool them all together, the identified aggregate level information, and put them together to get a better, more generalizable idea about like evidence about a question. So that creates a lot of opportunities for standardization. This is from data structure, how the data is stored, what type of data, the data content, the vocabularies that identify a specific domain, the semantics, how we define the disease population or specific covariates, like what is my study population, what is the outcome, the analysis, right? So that can be standardized, that's what you're doing, and also results reporting. So all of this, as long as we can standardize all of that, that help us move forward and get closer to where we want to do scalable generation of reliable and reproducible evidence. From the standard content, like from the data model, we have the OMAP common data model. It's a patient-centric model. I'm not going to go into that. We have standardized vocabulary. These are the components of the vocabularies from disease, conditions, drugs, observation procedures, and so on and so forth. Within Odyssey, we have Hades, which is a suite of standard methods library, which allows like standardized like analysis of different questions that can be a very deep like characterization of a specific patient population, looking into incidence rate, looking at assessing comparative effectiveness or safety of a drug versus the other, and also performing patient level prediction, along with a set of tools that enables like putting the evidence together or enables any of these analysis. So we're talking about this. Sounds really good. We have it all. We've done it. There are a lot of examples that you might have heard in terms of how Odyssey have made a huge impact. The most recent one is a lot of the work that has been done with COVID. But the question is, why am I here? Like, why are we talking about oncology as a completely different entity when compared to the rest of the medicine? Let me just give you an example. There's three problems that we've identified. The first one, cancer is actually a rare disease, if you think about it. This is a drug label, an FDA label for capmatinib, which is a kinase inhibitor for the treatment of adult patients with metastatic non-small cell lung cancer, whose tumor has a mutation that leads to MET exon 14 skipping mutation. So this is the population that I want to look at. Like, so if you think about it, lung cancer is the most frequent cancer and also the most common cause of cancer death in the US. Every year, we see around 200,000 cases of lung cancer, but not all of them are non-small cell lung cancer, around 200,000. Not all of them are metastatic, only 70% are metastatic. Among those, only 55% have adenohistology. And then looking at the specific mutation of interest that make those patients suitable for that specific drug, capmatinib, we're talking about around 1% of the entire population. So from 230,000 cases, all of a sudden we're looking at 7,000 cases. And the reality is actually way, way more complex because we're seeing co-mutation of like a lot of different genes that occurs, like patients have to go through several rounds of chemotherapy or treatment. So when we start like cutting and slicing this big pie of lung cancer or breast cancer or colorectal cancer, at the end of the day, we're seeing very, very small pieces of patients that are suitable for a specific drug or are suitable for a potential research question. Problem number two is that in order to do cancer studies, you need to have way more detail compared to the rest of the medicine. Common question that comes quite a lot when we look into oncology within pharmacoepi is, what's the overall survival or the PFS of patients with metastatic non-small cell lung cancer with confirmed MET exon 14 skipping mutations who have received oral capmatinib as first-line treatment. So for that, I need to know that a patient has a tumor that is lung, so the location of the tumor is lung, but a histology of non-small cell lung cancer, not every lung cancer, non-small cell lung cancer. It's not any non-small cell lung cancer. The tumor has to be metastatic. So I need to have information on tumor attributes. It's a metastatic disease. I also need to know the genetic biomarker information to be able to identify whether there is a MET exon 14 skipping mutation or not. Also, I need to be able to identify the regimen that the patient was put on right after the metastatic disease. So both the treatment that the patient is getting and the type of treatment. So the episode of the treatment and the regimen. So much more complex and like in a lot of detail in the data that we just generally don't see in the data. So looking into the data set, that's the problem space terminologies. How do we find them? Give you an example of non-small cell lung cancer, just looking at a histology of the tumor that's located in the lung that is metastatic. If you look at different ontologies going from SNOMED to ICD-10, LOINC, and NCIT, you see that these hierarchies are not complete. Not all of them have non-small cell lung cancer. They're not connected. So you might have non-small cell lung cancer, but you don't have the other one. You might have the metastatic disease as in SNOMED, but you don't have the non-metastatic disease if you were to study that. Not all and also in terms of the hierarchical relationship, you can't find the relationship between like all of the potential combination of only these three first attributes. Looking into lymph nodes, it's actually way, way more messy. You see like completely different representation of, for example, lymph node status from LOINC, SNOMED, CAP, or NACER. Putting them together or making sense of them is like something that is extremely painful for folks who have done it, and also none of them can comprehensively represent like any potential lymph node status that you need in order to be able to do a study. So we talked about the problems, and this is pretty much why we started around 5 years ago, I think, talking about it. Like, let's do something, because we were all like a bunch of people very interested to do observational cancer research. We want to use the data. We find it very difficult to do so. As I said, 3 big problems. Cancer is rare. It's very complicated to identify cancer patients in the data. And also we have terminologies problem. We cannot correctly, there's no terminology that can comprehensively define all the characteristics of patients. So we try to address them one by one. The power of network and what Odyssey actually gives us is enabling network research. So thinking about like all those small pies that are for the patient that I show you, that specific patient population, if I do it at each one center, I can only see a very, very tiny pieces of information or very tiny like number of patients. But within Odyssey and within the standardized method, I am able to put them together to get a better understanding of a larger, much larger patient population. So for some of these, sometimes we're like, if we are lucky, we can find 50 patients in a database. What 50, getting a reliable evidence is always questionable, especially if you want to like try to look at like the different combinations and different categories. Being able to look at it within a network would allow us to do way more than what we can currently, what we can do at the moment. The next problem was the complexity of identifying cancer patients, specifically like the level of details that we want, and also how the representation of this information within this common data model. So that's where we pretty much introduce Odyssey oncology module, which based on like the work that we do, we believe that it solves all of the problems within the oncology research in the observational side. So the module has 3 components: cancer disease model, how we define the disease. It's generally, I'll explain it later, a combination of a base diagnosis, where is the tumor and what is the histology of the tumor, plus a set of modifiers or attributes that would fine-tune that diagnosis to a specific patient population, biomarker, stage, grade, and so on. Cancer treatment model. Of a lot of time, like what we see in the data, like that comes from like EHR claims, is just like a single like administration of a specific drug. Cancer drugs are usually administered in combination with specific dosing and scheduling. They're called regimen. We introduce a composite level of information called treatment episode to be able to allow to represent cancer regimens and lines of therapy, in addition to ingredient level information that's already available in the standard OMA. And finally, the episode model. So when we talk about the episode, we're talking about continuous period of disease or treatment that have a very, very specific meaning. Metastatic lung cancer has a very distinct meaning. Localized or lung cancer has its own distinct meaning. These episodes can be composed of multiple events, and they're essential for conducting observational research. So going into that, talking about the cancer disease model, so the representation within the oncology module, that's going to be pretty much, that's our recommendation for doing oncology cancer, is to identify condition based on a combination of histology and topology, similar to what ICD-10 does. And then we have a set of diagnostic modifiers that can be tumor stage, it can be grade, can be margin of the tumor, the extension, whether there is metastases or not, the biomarker information, and these combination of any of them or all of them, in addition to histology and topology, would pinpoint the specific type of the cancer that we want to study. The cancer treatment model, as I said, allows for higher level representation of treatment and oncology. So thinking about like our patient that we're talking about, metastatic non-squamous non-small cell lung cancer, patient gets diagnosed, and then at some point, like a little bit after what they're put on cisplatin gemcitabine regimen, 21-day cycle for up to 6 cycles. So in the perfect world, you would see something like that, like how cisplatin and gemcitabine are co-administered. And this is what you would see in your data. And the best case scenario, which we know that it doesn't necessarily happen, the patient might not feel well. Day 15 or day 8 of gemcitabine might be a Sunday, so they're going to come on a Monday, or they're traveling, they come later. So that's like, it's not going to be as perfect, but this is what we see. In practice, this is what we want. We want to be able to identify a regimen based on combination of different anti-neoplastic agents, the number of cycles, and the doses, right? And be able to say that, okay, like which patient are getting what and how they are responding to specific treatments. So the oncology module enable representation of both regimens and cycles and also lines of therapy to be able to do that, in addition to supporting like extraction tools that generate information from drug level or drug exposure into regimen and cycles. And finally, the cancer episode models. As I said, episodes are continuous periods of disease. When we're talking about it, and they can nest within each other. They're essential to do cancer research. So we always start with a parent episode, which is the disease: lung cancer, breast cancer, prostate cancer. And each of them can have children, like dynamic disease and disease dynamic and disease extent, that I will explain. What's very important about that is that this is what the model allows: representation of different episodes of care, treatment, and also disease. Sometimes it's available. We can extract some of this information from cancer registries and we can populate the episode. In most cases, it's not available. Like an EHR, especially if it's not an oncology EHR, does not have that information. Claims data do not have that information. That should be algorithmically derived, or using NLP, machine learning, or other approaches that can derive these and then populate the episodes. So when we're talking about the episode model, just thinking about like a specific patient journey, it's a schematic journey. Patient is diagnosed with the disease. At some point, they develop metastases. Somewhere along the line, they do this biomarker evaluation. They go on their NGS panel. They were put on first-line treatment. There is a progression event. First-line treatment is discontinued. A little bit after the second-line treatment, you see another progression episode, discontinuation, and this can happen. This is just one example until the end of life, and like hospitalization, death, or anything else. So this is what we see. When we talk about the episode, the time from diagnosis to the end of life or end of observation is what we call the parent episodes of lung cancer. So we can just say that, like, well, from this time to this time, we have a patient has been diagnosed or has lung cancer. When the patient is diagnosed with metastatic disease, the extent of the disease, the extent to which the tumor has spread, and the body is changed to metastatic disease extent. So that's what we call the disease extent. Along the entire journey, we see periods of tumor progression. From a tumor that was not metastatic, the tumor is actually becoming more aggressive. We have a metastatic episode here. Somewhere here, there is an evidence of progression, and also here. So we see a disease dynamic of progression along this line, so at different time points. And then When we're talking about treatment, as I said, the patients put on first-line treatment and at some point they discontinued. This period is a first-line treatment of this specific patient. This is their first treatment episode and the second treatment episode. So when we talk about the episode model, this is what the episode model would do for a specific patient journey. You turn it into specific pieces of periods of disease with very, very distinct meaning. What's important is that with that, we're able to answer a series of questions that are very important in oncology, looking at overall survival from time of diagnosis, from metastatic disease, OS from the progression, looking at lines of therapy, looking at progression-free survival, because I have episodes of progression in this model, and a lot of different types of questions, time to next line of treatment, time to treatment discontinuation, having information on biomarkers, and also any of these episodes would allow us to also look into predictive and prognostic value of specific biomarkers. So that was the model, but the model also needs terminology and standardized vocabularies. So as I said, the first one was the disease, the cancer diagnosis. So the base diagnosis, if you remember, was a combination of topology and histology, very similar to how ICDO is representing cancer. So we've incorporated our ICDO to the standard vocabulary to SNOMED, and where applicable, like if there is a direct one-to-one match, they're mapped to SNOMED, otherwise they're within the hierarchy of the SNOMED conditions. For diagnostic modifiers, we're talking about stage, grade, biomarkers, tumor margin, the extension, so on, nodes, and all that. We've developed a set of vocabularies called cancer modifiers, and they're coming from different sources. So there has been like a series of investigation about like the most comprehensive vocabularies that are out there that can become standard for this representation. If available, how can we use it? And when it's not available, how can we actually enhance some of the current existing vocabulary? Specifically for genomic information, we've developed OMAP genomic vocabulary. I'm going to talk about it a little bit in a bit. For representation of treatment episodes, we're using HemOnc. This is a publicly available knowledge base of all the oncology approved indications for different diseases. The information is constantly being updated and coming from the NCCN, ASCO, ESMO, and the other guidelines. And for the episode model, the new vocabulary were introduced into the OMAP standardized vocabularies. I want to talk a little, yes.
I'll just go back a slide. This is such a great slide. There's a question, but I like to think of it in terms of structure and semantics. And semantics, it looks to me like, vocabularies, adding new terms. OMAP is such a flexible model for adding new vocabularies like ICDO and HemOnc. But then I think what's really profound is what you're doing in episode is structure. When I think of OMAP, I think of like pointillism. We have all these little points of data about the patient, but really the only groupers we get are it's this patient and it might be tied to a visit, but visit usually is a poor grouper because of the way it's implemented in EHRs. And this allows us to have a structure to say these points of data are part of this episode, just part of this staging of this cancer. So I think this is really an incredibly profound and fundamental component will allow us to do much more than oncology, but being able to connect the dots literally of our data by being able to say, here is, we can define these as periods of time around stages of disease.
That's correct. No, I think I absolutely agree with you, Paul. And this was pretty much the majority of the time that we've spent. I have to say that identification of the vocabularies and the structure and the mappings and what means what and how are we going to use it, that by itself has been a huge amount of effort, special support from the vocabulary team. But this piece and coming to agreement of the structure and representation, we're not saying that it's perfect or that is that's it and it should not change, but being able to represent the data in a way that allows us to do oncology research has been the core principle of everything that we've done. So we see the episode model as the essential piece of the oncology model, but I agree with you. This is not an oncology thing. It can easily be expanded to other diseases or other questions. I don't see the question. Should I look at it?
No, that was great. And you're right. I mean, think how easy it would be for doing pre-surgery and post-surgery, or pre-diabetic and diabetic, or epilepsy with relapses, or multiple sclerosis. I can, really literally any clinical, I could just see it is such a useful structure to be able to use. And so I'm really excited to see it being used in oncology, and I'm hoping to see it outside of oncology too.
So I think, to your point, there are currently effort from the other parts of the Odyssey, like we've been reached out, like disease-specific entities, like multiple sclerosis as an example, that there is a need for representation of different states of the disease. It's not oncology, but it's the same sort of concept that you need to be able to define and then define these different pieces or episodes. So there are certain efforts in terms of like trying to expand this to other conditions. So that, as you said, it's going to happen. Like we've been working on it and like we're still building, but it's also great to see that it's being used elsewhere.
Excellent. Thank you.
Okay. So I want to talk a little bit about the genomic variants and like generally how we're representing genomic information in OMAP, because within oncology, specifically the whole area of precision oncology, the genomic information is essential to do everything, right? Like used to be back in the day when lung cancer, at some point, then it became important to identify small cell, to be distinguished small cell from non-small cell, then adeno from squamous cell and all that. Then you see this rapid change that right now, like the disease are defined by co-occurring mutations or specific mutations in addition to histology and all that. So we need it, right? So, but the problem is that the genomic information is coming from NGS panels. They're not features. Like within the observational epidemiologic world, we need features to be able to say that like this present or not, then apply it like every other covariate, apply it like within like 2D code set to the study and do that. So what we need to do is turn these genomic features into an analytic data set. So just an example. Like a result of an NGS panel for a skipping mutation can give you information like this. It's a splice region variant at chromosome 7, this specific location. Exon 14 is missing, and that is the location. This is what we get from the patients. Ideally, that's what we want to have. So for patient 1, that specific information is not present. They don't have this. So like all of these features are specific genomic variants. So we will have 0 and 1, like present and absence. So that then become like an analytic data set, like any other ones, and we can use them. So a huge effort that we put a huge effort into identifying like what is the best way to do that. And like where there's like a lot of discussions about like what we're representing, how we're representing, and why we're representing that. So the goal and principle, and the first goal of the oncology work group has been focusing on enabling observational cancer research. So making sure that we can do an epidemiologic study using the already available information, right? A part of it is genomics. So that's one piece. Hopefully further down the line, we can think about discovery and genomic research, but our first and primary goal has been on making sure that the genomic information is integrated with EHR data or claims data to be able to enable answering a series of questions. So we introduced a new vocabulary called OMAP genomic. At the moment, we're looking around 80,000 variants within over 600 genes that are represented. So this information comes from very, very different knowledge bases. What we've encountered during this whole journey is that not one single knowledge base or available out there can represent everything, like the entire comprehensive cancer genomic information that's available and you might need for your research. You might get information from CIVIC, there are information in LOINC and CAP, and like other sources like NCI Thesaurus has genomic information, and also like genomic specific knowledge bases like CIVIC or CGI. or cosmic. So in the first step, we actually like incorporated the information that are coming from all these different knowledge bases to be able to come to put together a set first version of OMAP genomic with representing 80K variants. But the question is that, okay, like we have that, right? Like we have 80K, is it enough or not? Do we have it? And that's something that we've been dealing for the past couple of years, past 2 years. So just to give you an idea, based on like the latest publication, over 80 million variants has been identified in cancer. So 24,000 tumors have been sampled, 38 different tumor types. We're looking at 80 million variants that have been that's out there, right? Do we actually need them? Are there all like clinically important, relevant variants that needs to be represented in the data? Some of them can be passenger mutations. Some of them can be like a mutation due to age. Some of them are really like just like random findings that are not important for what we want to do. We also know that around 1,300 variants are known to be like pretty much like established drivers of cancer, coming from 680 genes. That seems to be way too narrow. We know that there are a lot of like other mutations that have been specified that are not a part of these driver mutations, but they play a role, prognostic or diagnostic. They have a prognostic or diagnostic significance. So that's what we're looking at, like this middle circle to be able to do a series of these questions, meta-exon 14, ALK mutation, EGFR, anything, or even looking at like composite level information, TMB, and so on. And this has been like our journey, at least for the past 2 years, to come up with this comprehensive set. Comprehensive means good enough to be able to answer oncology studies. We've done a series of evaluation in terms of the like where we are in terms of coverage, how we can enhance it and not. So I'm going to use this actually as a marketing opportunity because we're presenting a new tool at Odyssey in 2 days. So it's like the website will come up tomorrow that allows for better representation of genomic information in OMAP. So hopefully we're moving forward from this 800K to a much more comprehensive set, and also enabling a lot of the users and data providers with the ETL and mapping of their data to the genomic vocabulary. So I talked about like the problems of the cancer like that we have in oncology, so how rare it is, how complicated is it to represent it, and the problem of the terminology. I also talked to you a little bit about the solutions, what we've come up with as a group. But I want to use this opportunity to show you how it worked so far. Like just give you a couple of examples of like what we've done, sort of proof of concept, to show like sort of the module in action. So our first proof of concept was a study on bladder cancer, specifically metastatic bladder cancer. For those of you who are aware, like know more about bladder cancer, it's quite common. When it comes to metastatic disease, it's not that common. So a lot of information about like what happens to people who gets treated, who goes beyond first and second line is not really like described at extent or in large scale. So in the first step, we thought that, okay, this is a use case. We can actually use it as a proof of concept to see whether the module will work or not, like with the different pieces that we have available, right? So remember, like we've introduced the episodes, we've introduced the vocabulary and also the treatment episodes and all that. But the other part is that we need to have those data to populate everything. But we have something, and I can just tell you a little bit. So for this proof of concept, we looked at 6 different sources of data, a mix of commercial claims, commercial oncology EMR data, and also academic data from the US, like all linked to tumor registries, so sort of enhanced EHR data, oncology enhanced EHR data, and also academic data from Korea. So a little bit of like broader, like from like geography representation, and also in terms of types of data, we're looking at completely different types. So in total, we were able to identify 11,000 patients with metastatic disease, which is quite big for bladder cancer. And we were able to even identify and represent more than 1,300 patients with metastatic disease who have received third line of treatment. This is extremely uncommon to see this many patients across a network. So using some of the capabilities of the oncology module, we were able to identify and represent characterized treatments, the regimens and lines of therapy across databases. So, but for example, you can see here, like within the 3 lines of therapy, pretty much similar distribution of the treatment, first line, second line, even third line. A little bit of difference in Korea. The numbers, the sample size within Korea was small, and at the same time, there might be like some differences in terms of the practice. We were also able to describe time to treatment discontinuation and time to next line of treatment, quite similar across the different databases, and also very similar to what we know in the literature, all reassuring that we're not off, like we're able to capture it, we're able to represent it, and we're providing good and valuable information. So after this, we got to engage with the European Association of Urology, the main organization responsible for development of guidelines for urological diseases in Europe, and specifically an IMI project called Pioneer that's focused on generating a lot of evidence for prostate cancer, different questions. They have like a list of around 60 questions prioritized based on the needs for patients and the availability of evidence, and they're doing studies one by one. So this is like a great collaboration of a lot of like different entities like pharmaceuticals, academics, just like volunteers from the community. We organized a study-a-thon. Very specifically, the idea was that can we actually describe a natural history of disease, but not any prostate cancer, the set of patients that we want, like that they don't get treatment because we want to avoid over treatment. So something kind of different that what Odyssey used, like generally has done a lot and is very good as, like looking at describing disease and comparing different diseases. We want to look at just the disease itself, natural history of disease, and we're looking at a group of patients that are not getting any treatment. They're also very interested to use that information at the bedside. So come up with a prediction tool to be able to identify which patient is more likely or would be a good candidate for no treatment, and which patient should be put on treatment. So in addition to like large scale characterization, the desire is to actually take it to the bedside. So we did a 5-day study-a-thon looking at over 1.5 million patients newly diagnosed with prostate cancer. Data is coming from 18 different databases, Europe, US, different sources of data, EHR claims, cancer registry. And again, like data providers are also like from academia, industry. This is just a snapshot of the results. I don't want to go through it. All the results are publicly available, but just a take-home message is that we were able to actually do a very in-depth characterization of this patient population, patients who are treated, who are not treated, getting or coming to a definition of what do we mean by lack of treatment or no treatment, and also being able to identify and characterize the outcomes, talking about treatment initiation after diagnosis, mortality, emergency visits, and initiation of palliative and curative treatment. All the results are available here in case you're interested to look around and play with it, or even like, there's always like a need for new collaborators with new ideas to take advantage of all these results that have been generated. But that was just the start. So that has initiated a series of questions. We went through a journey of something that hasn't been done, like looking at like a specific population, things that are not easily understandable, distinguishable within the data. So a lot of like publication posters and all came out, like, can we identify like treatment intent? Like, how can we like distinguish two approaches that are based on no treatment, but the different intent and all that? And this is just like what you see here is series of like the research that has been generated, like this just like has been started after the study-a-thon, using the same data model, using the same approach, and adding more to it. Like a lot of collaboration is built through that, like in terms of how can we enrich this information using different machine learning approach, and can we actually incorporate some NLP and so on and so forth. And finally, in the last one, that's an ongoing study. I'm just like putting it up here. I have a Hopkins logo. So we have a collaborator who has recently moved to Hopkins. But this is something on specifically on non-small cell lung cancer and looking at predictive and prognostic significance of liver metastases. So the idea is to do it's a very specific collaboration with University of Alabama, Odyssey, and NCI, with an interest to look at the characterization of these specific patient population and also being able to look to see whether liver metastasis is or can be a prognostic or predictive, has any prognostic or predictive significance for these patients. So we are with this specific study at the stage that we want to make sure that we're enriching our data to a level that we can appropriately represent, specifically when it comes to outcomes. So again, using this tribune to just call for collaboration. If anyone's interested with data or no data, just to be a part of it, please feel free to reach out. Would love to have you. And just want to summarize it in one sentence as the oncology module, specifically the episode model, enables observational research and in a network setting. We're able to represent, we actually this can help with like the number issue and also being able to properly describe these patients. So the foundations are there. Like we're able to do a lot more than what we normally do with characterization and estimation, looking at relative survivals, like the changes in incidence and prevalence, and looking at statistical cure, and any sort of like the need for benchmarking, looking at real world outcomes. A lot of the work that's been happening in the oncology world these days, maybe more often because of how the drug development, how different drug development is in the oncology field compared to the other fields. So there's a lot of opportunities. There's still a lot of work to be done. So we would love to have more people who are interested, who can help us move this forward. This is a never-ending journey. So please join us. And I'm done with the talk. So happy to take questions.
Asya, thank you so much. Please submit your questions via the Q&A or chat. I also polled some other folks. So one of the questions I was thinking of is, you didn't just bring in one new clinical terminology. It looks like you brought in about 6. And so that's really a massive undertaking. So I'm kind of I want to understand the steps in that. How did you get those terminologies to give you a source of truth in a format that OMOP could accept? Like, in the right way, where they have a data catalog or data dictionary. And then I'm also interested, did you have to you always had to pick winners of what pieces of their vocabularies you wanted to say was going to be standardized and which part you're going to say we're really not going to use, right?
That's right.
How did you, how much, that seems like a large effort. And so how much, what are the types of steps for doing that, for harmonizing that vocabulary?
Paul, that's actually a very good question. I'm thinking about it because it just resonates with a lot of long nights being up. But to be honest, the way we started in the beginning was this idea and how can we represent it? And it's not available and not possible. And then we have information from cancer registry and all that. But at some point I got to a point that, okay, we have to have a systematic assessment and evaluation of what's out there. So as you know, within Odyssey, we're not into the business of creating new terminologies. We look for terminologies that are best represented, representing specific domains, like conditions, measurement, and all that, and we bring them in. So we kind of had the same approach. Let's have a very, very comprehensive assessment of what's out there. How are things represented? Going through all the CAP protocols. Going throughout all the CAP protocols, I'm not kidding that we went through all the CAP protocols to extract the information. Same thing with NACER, extracting all the information from NACER and evaluating Nebraska lexicon, NCIT, and all that. So putting them together and then have a proper assessment of where are the strengths and limitations of each of them. A lot of it is in we have a sort of a review publication in Journal of Clinical Oncology that goes through the steps and shows the whole summary of the representativeness and the pros and cons of each of the resources. So that's pretty much how we started. What do we know and where are these information and what can we learn from them? HemOnc We were probably a little bit luckier because that came through another collaboration that we have, like Jeremy Warner, an active Odyssey collaborator. We've been collaborating quite a lot. He has been putting this together. So that was a little bit of an easier path for us in the sense that we were involved in parts of HEMONC and how HEMONC can be incorporated into the vocabulary. So it seemed to be a little bit of a natural like choice, like, okay, like that seems to be good. But with the evaluation of this to present everything, what are the pros and cons? So after that, and after putting it together, it comes to a point that, okay, like we need to come to a decision, right? What's the best and most comprehensive representation? So I think the case of genomic, for example, like we did this whole assessment of literature, of all the available genomic resources, of everything, and brought in experts from the genomic field, people who are working constantly on harmonizing genetic information, for doing genetic research, to get a better understanding of what's there and how we can use them. So it was like through a lot of like these like iterations, and then we iterated and reiterated, and then we learned, and then we had like new folks, for example, like Joe would join and brought a lot of new ideas. So we had to pause, think back, and then like redo, and then do like try sort of building it step by step. So each of these vocabularies, and then in terms of like what are the like how can we actually fit the vocabulary? The episode was the other one, like the very good example of it's a completely new table. Like, how are we going to do the connection? What does that mean? Is it going to break the model? So we have this constant conversation of like, our addition of specific vocabularies, are they against the model? Like, are we going to break the rules? Like, how can we do that? We get a lot of input from the vocabulary team, from the CDM. So it's like a constant back and forth and communication between teams, and also a lot of like labor work.
So how long did that take? You think that takes about a year to add a new vocabulary? Because I mean, it really is, to do it well, you have to do a lot of harmonizing with that.
So I think like stepwise approach, I can tell you like from the very beginning, like I remember 2 years ago, we're sitting like going through all the CAP and NACER and all that to come up like, and then like the mappings, whether they're correct or not, like, and then like checking it and making sure everything happens. So to have like a first one release. And the first one release was good based on like what we had at that point. And then it keeps reiterating, right? So like in terms of like right now, if someone looks at like the first one release of like how tumor stage was represented, it's completely different right now. Because in the beginning, we were just like looking at like what we had. We had, for example, AJCC8, and then we had like a very pre-coordinated representation of the stage and based on like stage, tumor, and also the AJCC with the version. So right now we've created this hierarchy to make it much more straightforward. So I would say that this is like a continuous journey, and we're just like, and as we do it and as we apply it, we'll learn more, and then we incorporate it. So we have collaborators that have like data from AJCC6, or even before that, we want to allow them to use their data. We can't just say that, okay, like that's it, because that's the most, I don't know why, but so it keeps changing. Genomic is a completely different beast. It's still a constant effort. Like we want to get like, we're having a new release very soon, but through each release, we're finding like areas of, because we have to release and then we have to test it out. Like, does it work? Where are the gaps? How can we actually fit it in? Because there is no proper representation of everything that we require to be able to do like precision oncology or looking biomarkers. And it's just a continuous effort. Like, I don't think it's sometimes more than a year.
You know me, what excites me the most is the episodes. And I'm interested in, is there, I imagine there has to be a lot of flexibility in the concept logic because we don't know if it's an 8 fractions. We don't know if it's, 10 fractions. We only see each treatment. And then, so we have to do some inference. So is it like a logic model where you're looking for certain concept codes within a period of time and looking like for gaps between them? I'm interested in, how do you develop the logic models to identify these the periods of episodes for treatment?
So here, so I said like the episodes can be nested within each other, right?
Yes.
So like,
Yes.
For example, the parent episodes have disease extent episode and disease dynamic as their children, right?
No, I'm interested in like first line and versus second line. I understand that medications are going to be changing, but even understanding the different, identifying if they, the actual, even the each treatment, delivery fraction.
That's a very good point, and that is another piece that that's again happening on one side of the oncology work group. How can we identify them, and how can we actually identify them accurately? Because you're right. What you see are like specific episodes of administration, like a drug exposure or a procedure. We need to put them together. So our first attempt, and this is that's why we're just learning how to do it better, was, and this is again this is what's normally happening out there. If that information is not available, you come with these rule-based algorithm. Let's identify the first anti-neoplastic agents. Anything that comes within this many days of the first one, usually 28 days or 30 days, then we just put them all together. We call them a regimen. That's quite common if you do a literature search of how people define regimen when the information is not available. And then there are sort of rules for gaps and specific, for example, if other specific rules needs to be applied. That's problematic. If your patient is perfect, it works, right? That's great. You see that, but we know it's not happening. Oncology patients are frail. A lot of them are getting other medications that can be supportive or can be an oncology treatment. Dexamethasone is a very, very good example. Dexamethasone is a treatment for multiple myeloma. Patients with multiple myeloma are usually old. They're frail. They might be using it for other purposes. So at one point, how can we infer that? And also the dosing and scheduling changes based on the patient's condition. Sometimes it's three a component regimen would be started sort of 2, and then the third one might be added. So all of that sort of alluded to a need to do another sort of develop algorithms that are available to do some sort of fuzzy matching to enable some of this uncertainty into the algorithm.
I would love to see. Yeah, it's fantastic inference. And so what I envision
would tomorrow.
Be something like a supervised learning where we actually know the care plan from some either NLP or from some other source, and then we're looking at the points of data and saying which algorithms is going to have the most robustness for picking that out of these individual points of data to be able to infer the plan.
So we have actually two different approaches that we're working on simultaneously at the same now. One is we're borrowing some of the idea that are coming from sequencing and sequence alignment to incorporate the temporality and all of that, and then build something. And at the same time, we're doing some sort of supervised learning. So one of them will be presented in the symposium tomorrow. And at the end, that's a collaboration again with some of the Odyssey members, and quite exciting.
I can't wait to see that tomorrow. This is also a great warm-up for tomorrow's Odyssey Symposium. And so with that, I really want to thank you for coming and presenting about this exciting work. I think there's just a lot of extensibility. This is a foundation piece for helping us study all kinds of diseases and really taking Odyssey to the next level. And so I want to thank you for coming to present. And this closes our Grand Rounds lectures. And we really hope to see people in person tomorrow in Bethesda, Maryland for our annual Odyssey Symposium. And so it's certainly an exciting time of year. And I can't wait to see some of these results.
Yes, looking forward to that. Thanks for having me. That was really exciting.
Great. Thanks again. Have a great day, everyone.